Hello, my name is Justin Tran.
And I'm Kenton Felmley, and we're in Biology 545, Evolution of Development. Our project is titled When It Went, Stage-Specific Modulation of Medusa Morphogenesis and Colony Patterning by the Canonical Wnt Pathway in Podocarina carnea. What, where, why, and Wnt, the basics. Wnt expression experiments have been shown to affect oral-laboral patterning of cnidarians. Alstrapolone is used to upregulate Wnt expression, while ICRT14 inhibits Wnt. In Hydra, treatment with alstrapolone was sufficient to cause tentacle formation throughout the polyp, as shown here in a picture from Reinhardt et al. 2004. In addition, other treatments with alstrapolone were sufficient to cause secondary oral structures to form in Hydra, as shown here in a picture from Hobmeyer et al. From 2000.
In the life cycle of Podocarnea, when two gametes join, it turns into a planula, which then grows into a colonial polyps, which are either a non-reproductive polyp or a reproductive polyp. The reproductive polyps reproduce asexually, and the medusa reproduce sexually. The reproductive polyps will then produce sponge, which will then develop into medusa. The life cycle of Hydra is very similar to Podocarnea, where the gametes join and the planula grows into a solitary polyp, but they do not have a medusa stage. And this figure is from a study from Sanders and Cartwright. It shows that the expression of Wnt occurs at the oral tip of the bud. In early stages of the bud, Wnt expression is absent, but later stages from middle to later is when Wnt expression is observed. When Wnt signaling is disrupted or downregulated, you could expect the buds to regress or no longer develop. This study will focus on Wnt expression in the medusa stage and at a colony level, since it has not been previously studied. Using this figure further assisted us identifying the stages of the buds in our experiment.
A colony of Podocarina carnea was cut into 3 pieces, each containing multiple reproductive polyps. Each of the sections was placed in a small dish and left for 24 hours to settle. After 24 hours, each sample was treated with either 5 micromolar alstrapolone, 20 micromolar ICRT14, or a control of DMSO, because alstrapolone and ICRT14 are soluble in DMSO and are a component of these drug treatments. 24 hours post-treatment, the samples were washed and placed in regular media for the remainder of the experiment. 10 polyps from each treatment group, alstrapolone shown on the left, and ICRT14 shown on the right, as well as 6 polyps shown here for the control group, were observed every 24 hours, and the developing medusa buds on each of these polyps were classified as early, middle, or late developmental stage. When treated with alstrapolone, the Wnt signaling pathway is upregulated. The developing buds classed in the middle stage of development displayed regression, while those in the early stage continued to develop until reaching the middle stage, at which point they began regressing. Some buds were far enough in development that treatment didn't cause regression, but the resulting medusa from these buds displayed aberrant phenotypes, as will be shown in later slides.
When treated with ICRT14, the Wnt signaling pathway is downregulated in our developing buds. The buds in the late development stage showed signs of regression, whereas the developing buds that are in the early to middle stages showed signs of growth, but then later regressed. Some of the buds that were far in their development stage did not show signs of regression from the treatment, but resulted in medusa with abnormal phenotypes.
"24 hours post-treatment with ICRT14, 3 medusa were observed, lovingly named Amber, Judy, and Katie. Amber and Katie both lack an oral structure. The absence of any other phenotypic variants in Amber and Katie suggests downregulation of Wnt in developing medusa is sufficient to prevent an oral structure from forming. 2 days post-treatment, a 4th medusa was observed and named Beatrice, shown in the top left of this figure. Beatrice did not display an obvious aberrant phenotype. All medusa were removed from the media in order to cross-link their proteins and place them on a slide for better examination. Sadly, during the process, all medusa were dissolved and proved unable for further examination.
Post-treatment in Ulster for 48 hours, 5 medusa were observed that were named Bri, Kaylin, Stephanie, Soyan, and Rachel, and in all 5 medusa showed an aberrant bell structure.
Five days after treatment with alstrapolone, which upregulates the canonical Wnt pathway, many reproductive polyps displayed ectopic or wrong-placed tentacles. Traditionally, tentacles only form at the oral end of the polyp, but these polyps displayed tentacles throughout the polyp shaft, as shown in this figure, tentacles here, here, and here. One polyp in particular displayed formation of a secondary oral end here, and here are oral ends, while another appeared to develop a second polyp at the oral end, resulting in a polyp that had grown on another polyp, also shown in this figure below. The first polyp is here, and then another polyp has grown off of it. Experiments treating Hydra with alstrapolone display similar induction of an oral end, as well as ectopic tentacles. But due to the lack of colonialization in Hydra, there is no similar induction of polyp off of polyp growth. None of these phenotypes were observed in the control group and suggests that the canonical Wnt pathway wields a role in tentacle development, specifically in the patterning of tentacles along the polyp. In addition, 4 days post-treatment with alstrapolone, the colony displayed a much greater number of newly formed polyps, shown as light-colored here, compared to the already formed polyps here in darker orange. These are the same colony, just from a different view. So you can see the more widespread effect that alstrapolone treatment had 4 days after treatment on the colony altogether. In experiments of Hydra, treatment with alstrapolone has been shown to be sufficient to induce oral end formation, comparable to this induction of polyp formation displayed here.
7 days post-treatment with alstrapolone, several stolons, which are connective parts between polyps, were observed to have developed vertically rather than their traditional horizontal growth pattern. More experiments would have to be conducted on the role of Wnt in the directional development of stolon, but these observations suggest upregulations of Wnt induces vertical growth.
Overall, it appears that disruption of the canonical Wnt pathway in Podocarina cornea causes regression of middle to late stage developing medusa buds, while there was not an effect on early stage buds until they had progressed further. This supports the findings of Cartwright and Sanders concerning the stage-specific expression of Wnt in the developing medusa bud, and displays results suggesting a relationship between stage and treatment-induced phenotype. In addition, late-stage buds that were released as medusa displayed aberrant phenotypes, suggesting Wnt plays a role in the development of oral structures as well as bell formation. Upregulation of Wnt was sufficient to cause ectopic tentacles to form throughout reproductive polyp shafts, as well as vertical stolon growth and induction of polyps.
We would like to express our sincere gratitude to Dr. Cartwright, Dr. Heilman, Matthew Traver, and Amanda Ketzer for their knowledge and guidance throughout the course of our project. We specifically would like to thank Matthew for working with us every day diligently and continually offer valuable insight about our experiment.
Thank you.
Thanks.